My name is Frank Czerwiec. That's the Americanized, bastardized pronunciation. It's actually Czerwiec. I'm the CMO of Goldfinch Bio, a Cambridge-based kidney medicine company. And you see both the title of my talk and our business goal is to bring precision medicine to the kidney diseases. So we're very aligned with regard to what I'm supposed to be speaking about today. So let me get right into it. Sorry, let me see. This is just a brief description of how we do what we do. Our company is based on a product engine or platform that includes a kidney genome atlas, which means that we've been collecting DNA samples from patients with rare glomerular diseases, proteinuric diseases like FSGS, and now have, with controls, over 20,000 individuals and are performing GWAS and other analyses to identify potential targets for these diseases. And we are progressing with our partner Gilead in sequencing an additional 80,000 diabetic patients, half of which will have kidney disease, half of which are without. So those are the controls. And again, we're using the platform, we're using the analytics and computational biology to identify potential targets. We then move these into a human biology platform. So we do animal studies, but we go beyond that by using human iPSCs, either in culture, monolayer culture, or in organoids, kidney organoids, to test the various molecules that we'll be using to attack these particular diseases. And this helps give us a step of validation, which we think is really quite critical, because we know how many drugs have failed, or I'm sorry, have been successful in rodents, but fail in humans. And then the third pillar is a world-class chemistry group, which helps define molecules that meet the drug-like characteristics that we need to target these systems. We don't mention the fourth pillar, but that's where we are now, and I'll get into it a little bit. That is the clinical development, the execution and prosecution of how you do studies with a precision medicine perspective. So let me focus a little bit more on FSGS, which is the disease we're studying first. This is a rare orphan disease. It affects about 50,000 US and EU patients. It's very heterogeneous. And so precision medicine, in terms of understanding what is causing the disease in a particular individual and how to treat it, is important. Currently, there are no approved therapies for this disease. Everything that's being used are repurposed, already approved drugs like ACE inhibitors, ARBs, steroids, and more toxic drugs like calcineurin inhibitors. That's not appropriate. And basically what doctors do is they test something. If it works, great. If it doesn't, they move on to something else. Not an ideal situation for someone who is treating their child with this disease, as an example. So we're really focused on identifying a precision-based approach to treating this disease. A little bit of biology. I apologize for those of you who thought you'd never see these things again. But in this cartoon, we describe the filtration system of the kidney, which really relies on a cell called a podocyte. And you see on the right, on your left-hand side, what a healthy podocyte might look like in a cartoon. It has these foot processes, that's the word podocyte, that extend and cover like a filter the glomerular basement membrane, which is another part of the filter that helps the blood that would be coming from the bottom filter into the urinary space. The problem is that in some genetic causes and many environmental causes, the podocyte cytoskeleton becomes disrupted. And this leads to podocyte effacement, which basically means that the cell is retracting and losing these foot processes, and it can actually undergo apoptosis, die, and you lose the whole cell, which means there's a big gaping hole in this filter. Eventually, these things sclerose and you lose the whole nephron, but that's the issue at the core of this disease. And it's been known for some time that there are a number of systems that are involved. A very important system is the RAC one system. And this is a modulator of cytoskeletal remodeling. It's important for allowing the cells to flex, but not flex too much. And it can be activated by angiotensin, by high glucose or hyperglycemia, and by a number of other factors and toxins. And when it's activated, it actually causes a normally intracellular and developmentally, not needed calcium channel to be exported to the cell surface, where it can in fact allow more calcium influx, which further stimulates RAC1, and you end up getting a vicious cycle that leads to this cell death. And so while there are some drugs that affect this system, they are generally toxic, and the toxicities rely on the fact that they affect other cell types, and I'll talk about that a little bit more. But we're targeting with our drugs this TRPC5 calcium channel specifically because it's really only expressed developmentally in the kidney and the brain, and really is only used or useful seemingly pathophysiologically in this disease. In other words, I'll show you that the whole system is upregulated in patients who have FSGS and diabetic nephropathy. So how do we do personalized medicine? How do we approach it? First of all, you have to have a biomarker or some marker of the disease that can be used to select patients or select patients likely to respond to a particular therapy. And we're fortunate in that the FDA has paid a lot of attention to this in recent years with regard to proteinuria and other surrogate biomarkers in kidney disease. And it's really undergoing a renaissance, much like oncology has, where the importance of these biomarkers are recognized and are actually being accepted. So FDA now has accepted proteinuria as a biomarker for a number of diseases, including FSGS, IgA nephropathy, etc. And this article by Eliza Thompson, the Deputy Director of the Cardiorenal Division, highlights what their considerations are in terms of full or accelerated Subpart H approval. Basically, it's reduced proteinuria, but keep an eye on GFR. GFR is a measure of renal function. So if something is harming renal function, it doesn't matter that you prevent proteinuria. You want to see both of these changing in the same direction. So we'll get to that later. I had mentioned that there are some drugs that are used off-label for FSGS, and cyclosporin and certain other calcineurin inhibitors have been known for quite some time to be highly effective in the disease, at least in terms of proteinuria reduction. And the graphs that you see here are the results of patients being treated in addition to standard of care with cyclosporin A. Now, at 12 or 26 weeks, you see a high percentage of patients actually responding. The black bars indicate complete response, which is going all the way from nephrotic range proteinuria to normal, and partial responses in the gray, or for placebo in the white. So as you can see, these drugs seem to be highly effective. The treatment in this study was for 52 weeks. So at 78 and 104 weeks, you see there's even a persistence of effect, once you stop the drug. But why would you stop a drug if it's effective? I'm going to get into that next. And that is, this more recent study focuses on, again, cyclosporin A in contrast to another standard of care, which is another immunosuppressive agent. And while there are important changes in urine protein creatinine ratio, that's on the left-hand side, you can see that there are reductions by 26 weeks as high as 70% in patients treated with cyclosporin and somewhat less for mycophenolate dexamethasone. Those benefits in terms of proteinuria are accompanied by detriments, by actual nephrotoxicity, which you see on the right-hand side. So while mycophenolate dexamethasone isn't as effective, it also doesn't have any negative consequences with regard to GFR change over this 6-month period, whereas cyclosporin had about a 20% reduction in GFR. That makes it a problem and really a drug of last resort for this disease. Why is this? The next slide shows that both that cyclosporin affects calcineurin both in podocytes and other cells, and I'm using T cells as a model here because it was actually approved as an immune modulatory agent for transplant and other reasons. And calcineurin is important in podocytes, but it's important in so many other cells like pancreatic beta cells and the endothelial cells and smooth muscle cells lining the kidney, that it actually, those other off-target effects lead to its toxicities. So that's why you can target the same pathway, the same system more selectively if you identify something that's really only useful or expressed or overexpressed in this case in the podocytes. Our goal here is to suppress the activation of this whole system in podocytes and keep them more normal. So what happens when you block TRPC5 at the top of that pathway? In animal models, and I'm showing here two animal models, on the left-hand side, the docosalt model for FSGS, and on the right-hand side, a ZDSD rat model for diabetic nephropathy. In either case, there's a dose-dependent reduction in the levels of proteinuria that you see in these animals, which is important. We're not seeing changes in renal function, but it's hard to measure those changes in renal function in rats. It's really going to depend on what we see in humans for this drug, but we at least know it in principle, we're having the desired outcome in animals. As a company, though, we want to rely on human results. And so what you see in this next slide on the top are human iPSC-derived podocytes grown in monolayer culture. On the bottom row, human kidney organoids, which include all of the components of the glomerulus and proximal tubule. The very far left is the control or baseline situation where actin filaments are all engaged appropriately. When you add protamine sulfate, which is an activator of EGF, this actually causes these actin microfilaments to become aggregated and dysfunctional. So that's sort of the feet of those podocytes pulling up and becoming damaged and leaky. But when you add the drug, you see that can be reversed or stopped. And the small graph on the far right shows it in a dose-dependent manner. So we know that activation of the system is important. We know that blocking its activation seems to have some potential benefits, at least in terms of the modeling. How do we convert this to a personalized medicine? What you see in this slide is a proprietary urinary biomarker that's been developed by our company over the past year that really measures the activation level of TRPC5-RAC1 in the urine. On the far left, you see normal and DOCA-treated rats, that there's a difference between the level of this biomarker in those conditions, and healthy humans and patients with diabetes, FSGS, or Alport's disease. Importantly, this is elevated in DN and FSGS. This biomarker is not elevated in Alport's or healthies. And what that means is that proteinuric diseases, like the last three, DN, FSGS, and Alport's, all share proteinuria as a biomarker, but it's not relevant to the disease mechanism. And so when you inhibit the TRPC5-RAC1 pathway with this molecule, you see the positive effects in the ZDSD diabetic model, the docosalt rat FSGS model, but you see no effect, no changes or benefits in terms of proteinuria in the Alport models. So that's internally consistent in terms of mechanism. Now I'm going to turn a little bit away from our drug and talk about biomarkers in general for kidney disease. As I said, proteinuria can be used as a biomarker. In fact, it's one of the most commonly used biomarkers in addition to GFR or renal function. And one of the very recent successes in renal disease is the approval of canagliflozin for not only improvement of all outcomes, cardiovascular, mortality, et cetera, but more specifically for renal outcomes. And the first study, Canvas, focused on safety and outcomes for patients treated for diabetes, but they were careful to look at the data, and you can see that different levels of proteinuria, in this case, urine albumin creatinine ratio, lead to different levels of efficacy in terms of reduction of that proteinuria. In fact, you see efficacy across the board, but it's more marked and more evident for patients that have high levels of proteinuria. And that also translates to benefits that we're seeing for GFR on the right-hand side of the graph. So you see that there's a greater spread or greater ability to detect those levels. And it's more important too, because those patients are declining in terms of their renal function much more quickly. So in fact, this biomarker was used as a way of enriching the subsequent Credence study, which is the one that was used to show that the drug is beneficial for kidney disease. Another use of biomarkers is described on the next slide, and that's the SONAR study, where you're using biomarkers to select patients. And here they used not just baseline proteinuria, that was an inclusion criteria, but they also used the reduction in your baseline proteinuria as a criterion for enrolling patients in the study or determining whether the patients might be differentially benefited by this endothelin 1 antagonist. And so they had a run-in period where everyone received the drug. Part of that was to test who were responders and who were non-responders. The other part was to see who had negative responses, because the endothelin antagonists actually cause edema, and in earlier studies had problems with patients developing heart failure or symptoms of heart failure. So they wanted to exclude those patients during that run-in period. So it's kind of an exclusionary test, but they also used the response of proteinuria as a means of enrolling patients. What you see here is that responders and non-responders both responded to the drug in terms of the overall outcome, primary composite being doubling of creatinine, ESKD, or end-stage kidney disease, or death from end-stage kidney disease, to more or less the same extent. The ends are different, so the power for detecting the differences is there. But basically, they said the drug could help patients, but you really couldn't count on proteinuria or the response of proteinuria as a means of selecting which patients are going to benefit. The best they could do is eliminate patients who are going to have harm. So in the final slide, I just wanted to outline where we are in terms of our program for 887 and FSGS and DN. And we are moving from a healthy volunteer safety PK study, which was completed last year. We did test the biomarkers, and healthies do have levels of biomarkers, and we are able to demonstrate target engagement using that, which is very beneficial for helping us move to phase 2, which is where we're beginning in the next few months, COVID allowing. And these patients will be FSGS and diabetic nephropathy patients. Will do PK/PD and dose range finding. And this proof of concept would hopefully lead us to phase 3, but it'll also give us the data necessary to further validate the biomarker. And in phase 3, in addition to using proteinuria as an entry criteria, because that is important for the different types of approval FDA might grant, we'll be able to enrich for patients who have activation of this pathway using the biomarker, and potentially identify whether those patients are more or less responsive to the drug. We will, of course, include biomarker negative patients. That's necessary to support the further validation of any companion diagnostic. And as a result, if all of this is successful, what we'll be able to do is say, this drug works for patients that have elevations in this particular pathway and should only be used in those patients because they will have the maximum benefit to risk for treatment. I'll stop at that point and entertain any questions you might have.